Welcome back to my channel Excellent proteinX. In this tutorial video I'm going to talk about the Ncbi blast, a protein alignment sequence using the Ncbi blast. So for that you have to type Ncbi blast and then search. After search you can get this one. So to click this one, basic local alignment search tool, and then after that you will get a redirect to the Xgene blast interface. So this is the DNA sequence for blast alignment, and this is for protein sequence alignment. So right now I'm gonna show you the protein sequence alignment. So to click this one, then you'll get this box for your desired sequence. You have to paste here. So usually you can see only this one first. So when you open it at the beginning, you will get only one box for pasting your sequence. So after that you have to click this one. So when you click this one, then you will get two boxes. Alright, so I already have one sequence, two sequences. So here in the input, in the spike protein I searched here. So there are two different spike protein I would like to align. This is the SARS-CoV-2 and this is the normal SARS-CoV. This is the coronavirus. So there are two different sequence I already have. So I can copy one by one. Copy and then paste here. So one sequence here and another sequence. I have to copy and then paste the second box. So after copy and after pasting all of the sequence, then you will do, and here also, yeah, after that you have to hit the blast button. It takes little longer time. So after that you get redirected to the alignment page, the result page. So you get this page. So here you have to click this on alignment. So after clicking this one, then you get the result of this alignment result. So here as you see, this is coil 1 and this is subject 1. So this is two different sequences that I pasted. So this one is the SARS-CoV-2 and this is the coronavirus SARS-CoV. And here as you see, in the result it's showing the identity is 76% and gap 2%. There are 1277 sequence. So out of that 26 gaps it's found, and out of 1277, 971 is identical. The rest of the sequence are not identical. So it is, if you look closer, here you see this in the upper there is V and here is I. So there are some places that is identical, it's showing the same, but when there is some mutation, then it's showing like V here and it's I here, and this dot, this dash, it means that it's deletion. So in this sequence SARS-CoV, there is one Q, there is one H and T, but in place of here there is no H and T, there is deletion. So it's not that there. And also you see here in this sequence. So here these are these tests. So this test means in this place there is no this sequence. Alright, so here you see if you scroll down further, then you get this 1273 sequences. Alright, so this is how you get the protein sequence alignment. And if you would like to do the DNA sequence alignment, then also you can do in the previous page, just previous one, you will select the nucleotide sequence, and also from here you can also do that. This is the nucleotide and this is the protein. So if you click this one, then this is the nucleotide. So it's the same, only you have to click this one, and then similarly you will get the two boxes, and you have to put your one sequence here and one sequence here. After that you have to click the blast, and no other settings need to change here. Yes, everything is as it is, no problem. Alright, so I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and subscribe my channel. Thanks.